Okay, we're on take 3. Yay! Okay, we are going to talk about eubacteria. I'm backing up just a little to a couple things we already talked about in class just to make it a good starting point for me to be able to use the video another time. We're going to be talking about eubacteria. Remember, there's archaeobacteria and eubacteria. Archaea are the extreme bacteria. Eubacteria are diverse, and there's a most numerous groups of organisms on Earth. A large variety of bacteria are needed in order for the Earth to work. And they did a project several years ago, like 20 by several, maybe longer, called Biosphere 2, where they created a biosphere that was supposed to basically make a mini Earth that had all the resources, and within that, you could live and survive within itself. It had different biomes, like a rainforest and a desert and those things. And it also had people, a living area, an agricultural area. The idea was that if you ever wanted to colonize Mars or the moon or something, that would give you an idea of what was needed to do that. It was not super successful, mostly because of people. Okay, so eubacteria. First thing about them is, how do they reproduce? They reproduce by binary fission. They double their DNA and then they split in half. Okay, double their DNA, split in half. Structure. What do these look like? They are 0.2 to 2.0 micrometers long. So super, super tiny. You could fit 1 million of them side to side on a yardstick. They are prokaryotic, which, if you remember, prokaryotic means that they do not have any membrane-bound organelles. They do have a cell membrane, but no membrane-bound organelles. They also form endospores. Endospores are a tough structure that protects a dormant bacteria. So, if a bacteria can't grow and reproduce, the conditions aren't right, it can go dormant, and it has an endospore that then protects it. Okay, it has a cell wall. Let me move you back. With the peptidoglycan. Remember we said that archaeobacteria did not have that. This cell wall provides some protection. It also has an outer cell membrane. So remember, a cell has a cell membrane, and then a cell wall would be outside that. This has another membrane outside of that. And that membrane is going to be so it can anchor to a surface. The outer membrane is so that it can anchor to a surface. It can also have a capsule. This capsule is very hard to destroy and is often pathogenic. Pathogenic, think the word pathogens. They are disease-causing. Okay? So, then they often have what are called fimbriae and flagellum. So, these fimbriae are thin structures that cover the bacteria and help them to attach. The flagellum, remember, are kind of like a tail. They allow for movement. Okay, so then we have the word normal flora. So flora always refers to plants. We always think of flora, and then fauna is animals. Yeah, okay. But flora, so bacteria. So normal flora is what organisms, let me put my words together, have in a healthy body. So your body has bacteria in it. Good bacteria, you'll hear it said. For example, one of them in your gut produces vitamin B12, which is good, and we need vitamin B12. They also compete with bad bacteria for food. So you have the good bacteria that eat some of the food and bad bacteria. Well, if the bad bacteria, the disease-causing bacteria, can't get the food, then the good bacteria can reproduce more and more and help keep you from getting sick. Does that make sense? Okay, so we have shapes of bacteria. So we have three shapes. The first is a circle, and it's called a coccus. The second is kind of an oval, and it's a bacillus. And the third is a spiral, and it's called spirillum. See if I spell it right. Okay, now there we go. So coccus is a circle, bacillus is an oval, and spirillum is a spiral. Now you can have these form chains, so you can get little chains. They can also form colonies, where a whole bunch of them are together. We do use the shape as a way to classify bacteria and try to figure out which one it is. Okay, biochemistry of bacteria. Okay, bacteria is a producer. So it doesn't produce via photosynthetic, although some can be photosynthetic. So it can be photosynthetic. It also, okay, so let's do the photosynthetic. So they're producers, so they can be photosynthetic. They do not have chloroplasts because they're prokaryotic and do not have membrane-bound organelles, but they do photosynthesis across the cell membrane. So they have the chlorophyll needed in the cell membrane. An example of this is called cyanobacteria. Another example is in tube worms. This is a non-photosynthetic. The bacteria can convert chemicals into organic matter, and that's called chemosynthesis. So you're taking chemicals and making food out of them. So photosynthetic, or example, bacteria in tapeworms, in tube worms, sorry. The bacteria in the tube worms can convert chemicals into food. Okay, now we have aerobic versus anaerobic. And hopefully you remember what those words mean. Aerobic, we have to have oxygen. Anaerobic, we do not need oxygen. Oxygen required, no oxygen required. Remember I said that when you're culturing, if it requires oxygen, it is better to do it on an agar plate because it will still get oxygen. Whereas if you do it in broth, it may sink to the bottom and not have oxygen. So we have three different types here. We have an obligate anaerobe. Obligate aerobe. That means they must have oxygen. The obligate aerobes must have oxygen. Then we have an obligate anaerobe. They do not need oxygen. And then we have the facultative anaerobe. This means it can do, facultative anaerobes can do aerobic or they can do fermentation, which is anaerobe. So obligate anaerobe has to, notice the word obligate, like obligation. Obligate anaerobe cannot use oxygen. Facultative anaerobe can use oxygen or cannot use oxygen. Okay, immune system. You have one of those. This is how we fight off bacteria in our bodies. So, our immune system is designed to keep harmful bacteria from overgrowing, from getting too much harmful bacteria. But we have to have a balance between good bacteria and bad bacteria. Even a good bacteria can cause problems if you get too much An example of this is E. coli. So you have E. coli in your bodies. But if you get too much E. coli, you will start to have problems. You also, the location of our normal flora can be a problem. So we might have bacteria on our skin that's okay to be on the outside of our skin. But if we get a cut and that bacteria goes inside our bodies, then it could cause a problem. So that's why one of the reasons we clean out cuts. Serology, and I'll write that up here, is the study of blood, serum, and immune responses. I'll say that again in just a second. Serology is the study of blood, serum, and the immune responses. So how are we responding? How does our body work to respond to these things? Okay, let's classify some bacteria. So one kind of thing is you can have strains. You've probably heard of this. So a strain is not like a whole new species, but it is a bacteria within a species that has just a slightly different characteristic. So maybe a strain of the flu, or no, it's not good because the flu is a virus. That's not a good example. But you can have strains. So you have the same species, but then you have strains within that same species. Okay? Okay, so how are we deciding these? And hold on, I want to make sure. Okay. So we have, remember we talked about the gram staining. So gram positive staining versus gram negative staining. So that's a big part of how we divide these up. So the first thing, I'm going to move this here so I can spell it. Proteobacteria. Gram negative. So gram negative proteobacteria. So as you can probably guess, they are gram negative. They're anaerobic, meaning they do not require oxygen. They have a low level of nutrients. There are 5 groups or classes within that. They also do what's called nitrogen fixation. And we will learn about the nitrogen cycle. They do what's called nitrogen fixation, which is they take nitrogen that's in the atmosphere and they turn it into a nitrogen that can be used by a person. And there's quite a few examples of this, and that's part of what you'll be reading in your assignment. So you're reading pages 208 to 218, and it talks about the specific examples within this. You are not going to be required to know a ton about each specific example, but just to understand there's a lot of variety. Okay, so we're going to do one example. Oh yeah, I'll move this so I can read. Rickettsia. These require a host. So they are parasites. So they require a host in order, so they are parasites. They use the human cells to reproduce. They don't reproduce just on their own cell. They use the human cell to reproduce. Then they move via flea or tick to another organism. So Rocky Mountain spotted fever is an example. So it's a little interesting. Maybe I'll find a graphic of that to talk about a little more. Kate may have more good information on that. Okay, then we have non-proteobacteria gram negative. So, I gotta erase again. Let's see how to spell proteo. Okay, so again, we are gram negative and they live in a diverse habitats. Again, I'm not going to go into all the specific details of them, and you're going to read about the variety that's in the book. There's a lot of variety of different organisms, and it's nice to read about them, but we won't necessarily remember every detail. Okay, and then the third class is gram-positive. And so this is the firmicutes. And so we have firmicutes and actinobacteria. Firmicutes have low cytosine and guanine. Remember when we make DNA, we need cytosine and guanine? They have low cytosine and guanine. They form endospores, so they're hard to get rid of. An example of this is botulism, which is a super deadly bacteria if you get it. It comes from things not canned properly. The actinobacteria has higher cytosine and guanine, and it has filaments that make a network-like appearance. Okay, again, you're going to read about some of the examples. Now we have a unique category that doesn't fit in anything because it's not actually alive, but it kind of is. It's not alive, and that is viruses. Viruses, viroids, and prions. Okay. Viruses, viroids, and prions. These are not alive. They must live inside of cells of other organisms and use that cell's biochemistry to work. They only have proteins and nucleic acids. The proteins form a protective coating. So it's just proteins and nucleic acids. Remember, nucleic acids make our DNA, RNA, those things. So they have proteins that form a protective coating, and they have the nucleic acids. A viron is an individual virus particle. So an individual virus is a viron. A capsid is the protein coat around a virus. So there are 4 types of nucleic acid. And these are, I don't know that you totally need to know them, but it's ssDNA, dsDNA, ssRNA, and dsRNA. This is single-stranded, double-stranded, single-stranded, double-stranded. Okay. There's a couple different types of viruses, whether they can go into a human or different types of animals. So we have bacteriophages. Bacteriophages. These could only invade bacteria. We have a plant virus or animal viruses or human viruses. And a lot of times they don't jump from one to the other. I'm going to give you a little bit about how viruses work, and we might do some more to make sure we understand it. Because once we've taught this, that is all we're doing for the quarter. So then the next few weeks, we can do more hands-on things to learn some of the concepts. But all of the basics have been taught. Okay. So maybe I'll see if I can find something that kind of acts out how it works. Okay. So it must attach to a target cell. And then it penetrates the cell. And it does that to get the nucleic acid inside. Okay, so we have 2 versions here. We have lytic and lysogenic. Lytic reproduces using the cell's biochemistry. So it just goes in and uses the proteins and things within the cell. Lysogenic inserts into the host genome. So it takes the DNA of the host, puts its DNA inside that DNA, and as the host cells reproduce, the virus will reproduce with those host cells. So I'm hoping that makes sense. So it takes a human, let's say a human, we're actually going to use chickenpox as an example for this. It takes the human cell, it inserts into the DNA of that human cell the chickenpox virus. Okay? Then that cell just goes about its life reproducing and reproducing and reproducing, right? And new cells are reproduced. That virus then can lay dormant, which means it doesn't show up. And it just lays dormant inside that cell. And then when a certain condition happens, it will trigger that to react, and then the virus comes out. So the example of a lysogenic is shingles. It can lay dormant, and then when a stressful event in life happens, it will then rear up, and you will suddenly have a viral infection of shingles. So it uses it literally inserts itself into the host DNA and stays there. The lytic just uses the proteins and things within the cell, the ribosomes and whatever, to reproduce on its own. Okay? So I will try to look for some things to help kind of cement that concept. But that is the rest of chapter 7.